Good morning, everyone. Myself, Dr. Sakshi Bhardwaj, on behalf of Altum Technologies, I would like to welcome you all and thankful that you could have joined us today for this next subsequent session of our Women's Day special webinar series. Today, session actually entitled as Development of a Computational Pipeline to Enable Network Pharmacology for Traditional Medicine. As we all know, Ayurveda, that is Ayu or life, and Veda or science, is regarded a science of life. It is also titled as Mother of all the healings, that is a longevity enhancing therapeutic science. There are two guiding concepts of Ayurvedic medication formulation. Actually, the one using one drug at a time and the another one using multiple drug at once. This using multiple drug at once is called as a polyherbal formulation. The idea behind the polyherbal formulation is that the herbs, when blend together in a very precise ratios to create the polyherbal treatment, they might have stronger medical influence or lesser toxicity while working on the multiple human body targets. Network pharmacology models have shown to be useful tool in explaining basically the molecular foundation of potential formulation mechanism. To explain the concept of polyherbal formulation with their pharmacological network, we have our speaker of the day, Dr. Manali Joshi, Assistant Professor Bioinformatics Center, Savitribai Phule Pune University, Pune. Ma'am has done bachelor's in microbiology from Savitribai Phule Pune University, master's in biotechnology from IIT Bombay, and PhD in biochemistry from University of Houston, USA. Ma'am also has an advanced diploma in bioinformatics from SP Pune University, that is the Savitribai Phule University, Pune. Ma'am had a work experience with the V Life Sciences Private Limited as well. Ma'am has her research interest in using and developing computational tools to look at various biological problems. Her primary interest is to understand the structure, function, inhibition, paradigm of proteins such as methyltransferase, kinase, GPCRs, etc. Ma'am has been interested in using computational tools to understand the underlying principles of Ayurvedic rasayanas better. Most of her work is done in collaboration with other experimental and the computational groups. Now, without taking much time, it's up to you, ma'am.
Thank you, Dr. Sakshi, for this kind introduction. At the outset, I would really like to thank Altum Technologies for this excellent series on women that they have organized. It is a great opportunity for all women who are trailblazers in their own respective fields to get this chance to talk about their work. So, as Dr. Sakshi has very elegantly given a brief introduction about what is Ayurveda and what is network pharmacology, I will take this opportunity today to talk about our most recent work, which I am very excited about. It's unpublished work as of yet, but I will try to convince you how we are trying to change how people will do network pharmacology in the future. So, today's talk focuses on developing a computational pipeline, like the title suggests. So, let me start by going back into history a bit. So, ever since mankind has been around on Earth, it is very difficult to place an exact time when people started looking around for remedies, right, for diseases or certain other maladies. And different cultures around the world, different groups of people, as they started evolving, they noticed that there were some solutions right around them. Okay, so we have these different um, groups across the world. You can say these were civilizations as they developed. They started having their own traditions. And you can see on this map, as is highlighted, almost all places around the world have their own traditional medicine systems which have evolved over years and years. And this has actually come from a very basic primal need of any person, where they want to find solution for any type of disease that they feel. They want to find solutions to, lessen the suffering. They want to find solutions for increasing longevity. And slowly this emerged and evolved. I think one of the oldest evidence one can find regarding people looking for solutions around, and especially one knows that most of the solution came from plants. So in some of the very oldest evidence found in Philippines and Thailand, in some caves where the use of uh, betel nuts or supari, which we know as, there were also some paintings and carvings which allude to the fact that people were using hallucinogenic mushrooms, okay, as some remedy. And as well, things evolved and people started the art of writing down and noting things. We also have different cultures with these old traditional manuscripts. So, for instance, on the top left, you can see that's a picture of the Ebers papyrus, or what comes from the Egyptian culture. So the Egyptians had, as you're all aware, lots of gods, and they had lots of medicinal remedies written as well. Then there is the most famous traditional Chinese medicine, which I would say is more recent, in the 16th century. And of course, like Dr. Sakshi has mentioned, we have our own Ayush system, and she highlighted Ayurveda, for instance. So I have here a copy of a very old scripture. So Ayurveda was written many years ago, 10 to 1000 to 2000 BC. So people believe it started from the Vedas, and then it was rewritten and slightly modified. And then you have versions in, of course, the Charak and Sushrut Samhita. So we know that across the world, these traditional medicinal practices have been there. But slowly in the 19th century, as science evolved, scientific methods evolved, there were so many revolutions in terms of biology, the physiology, medicine, anatomy. And slowly, Western medicine, as we know, emerged. And the way it has emerged is that we have a lot of scientific principles behind how things have evolved. Okay, and we'll talk a little bit more and compare and contrast and discuss why we need to do something more in terms of these traditional medicine practice. So while we have Western medicine, which is absolutely accepted and practiced from the 19th century, but we still have these traditional medicinal practices all around the world. So the World Health Organization has actually taken note of this, and there are a lot of efforts ongoing to actually take a detailed look at this and see what we can do better in terms of traditional medicine. So the way the WHO defines traditional medicine is it's the sum total of the knowledge, skill, and practices based on theory, beliefs, and experiences indigenous to different cultures. Okay, so like I had mentioned, it's not just our Ayurveda or Chinese medicine or Egyptians, it's across the world. There are different traditions that people have had, which have evolved over years and years, which is used for maintenance of health, prevention, diagnosis, improvement, or treatment of physical as well as mental illnesses. But the thing is that many of these traditional medicines in many countries are not mainstream, but they are now identified as complementary medicine. Okay, so the definition of complementary medicine would be a broad set of healthcare practices that are not yet fully integrated. Okay, and like I said in the past, the way things evolved over years and years, the first reports that can be seen historically is that people were looking at herbs. Okay, in fact, the way modern medicine also began, for instance, with morphine. So, morphine is derived from opium or poppy, as we know. Aspirin is derived from the willow bark, quinine from cinchona bark. There are so many approved drugs on the market who have their origin in herbs. Okay, so the WHO recognizes that herbal medicines are herbs, herbal materials, preparation, and finished herbal products that contain as active ingredients different parts of the plant or combinations. Okay, so like Dr. Sakshi had introduced, in fact, even in our Ayurveda, we have these polyherbal mixtures. So let us just keep this in mind that as of now, today we have Western medicine, and in many countries we have these alternative or complementary medicine practices, which are basically based in these tradition that have come from centuries and centuries. Now the WHO recognizes that this is there to stay. Traditional medicines are there to stay. In fact, a lot of countries are using so many of these as herbal supplements. So, why is this still complementary? Why is global acceptance still a problem? And why do we need to work on this? Okay, so one of the first, let's say, issues perhaps with using herbs, and I'll refer to herbs because although we have many other sources from which these traditional medicines arise, I think uh, coming from herbs is one of the biggest components. So let's talk about herbal principles. So the problem is that there is a lack of standardization. Okay, so for instance, ashwagandha had become a big rage. In fact, last year, if you look at social media records or YouTube, you will see that a lot of TikTok users also were talking about ashwagandha. Suddenly, it became extremely popular as an adaptogen, as an anti-anxiolytic, but there were also many others. So there were opponents as well as proponents. While a lot of people saw benefits, there were some others that warned about some other safety concerns. Now, why is this? Okay, this is because of lack of standardization. Now, being a plant, or right, it depends a lot on where the plant grows. So we have ashwagandha, which can have so many molecules that are extremely well characterized, right? There are these vithanolides, there are vidafferins, and so on. So there are categories of small molecules like alkaloids, steroids, flavonoids. Now the issue is that being a plant, okay, it depends on where the plant grows, how the plant grows, in what condition it grows. The concentration of these chemicals might alter. Okay, so today if you take a sample of ashwagandha root, is it going to be exactly the same as you would, let's say, from somewhere else at some other time, grown in some different conditions? Okay, so we really do not have control over what will be the exact chemical ingredient. So this is one of the biggest issues of plant-based medicine: lack of standardization. Okay, the other thing is safety concern. Now people are using a lot of these as herbal supplements. Okay, but we don't know how these interact with, let's say, Western medicines. In fact, we are beginning to understand. Now one of the celebrated examples is of this plant called the St. John's wort. So this is used quite a lot as a herbal supplement in Western countries. And people then realized that what St. John's wort does is that it increases the expression of cytochromes in the liver, and that means that it affects the metabolism of all the other drugs that a person might be taking. So when any person consumes a medicine in the form of a small molecule, the it will be absorbed, right? We hope that we are taking a medicine to be absorbed. So it then passes through the liver where a lot of metabolism can take place. So the dose that has been standardized for, let's say, average population use is standardized based on assuming a level of cytochrome function. However, now if you're taking suddenly some herbal supplement like St. John's wort, where it's going to increase the action of these cytochromes and it's going to increase the metabolism of the other drugs that you're taking as well, which means that the amount that will go into the bloodstream, okay, in the general circulation for availability will decrease. So suddenly people were reporting that, for instance, if they were taking St. John's wort along with contraceptive pills, they were seeing breakthrough bleeding because it was not as effective as it would. Now this may not be, doesn't sound right now like a very life threatening side effect, but it can be. For instance, if it's going to interfere with your heart medication, if it's going to interfere with your immunosuppressive drugs, and in fact St. John's wort has been shown uh, to interfere with all of these, right, by changing the expression of cytochrome. So this is definitely needs much more study. So we already know that when doctors give us advice that one can take herbal supplements, but under guidance of your primary care physician. So which means that for these herbal chemicals, we need a lot more study, right? How it's going to interact with other things, for instance, that you may be taking. And of course we have limited scientific evidence. So when western medicine evolved, right, there were so many advances that now we know minute details up to molecular level, we know, right? But the same is does not hold true for let's say Ayurveda or any herbal medicine. It is only now that we are trying to begin to understand what might be the components, how it might be working. So, to gain global acceptance, to be more confident about use of these traditional systems, and definitely there is something important there because people have been using these from centuries. There is a lot of experience there. Um, I mean, it builds on so many years and years of experience. So, we do need to put in a lot more scientific evidence to understand how it works and how best to use it. Okay. Now let's compare this with modern medicine. So, for instance, let's take an example of aspirin. Now, when we pop this pill of aspirin, we have this pure chemical, and you can see the 2D structure. We know that aspirin works by inhibiting the COX-1 and COX-2 enzymes. These are cyclooxygenases. And we know by virtue of this inhibition, we have relief from pain, fever. It acts as a blood thinner, and it's actually prescribed as a blood thinner post myocardial infarction. And it's used for prevention of clots to avoid stroke as well, right? So on the right hand side, you can see that we even know the molecular level details of how aspirin works. So it actually goes and binds at the active site. So at the active site, you have arachidonic acid, which goes and sits, which is converted to the product. Now, when aspirin is in the body, it will actually go and acetylate a serine. Okay, so that blocks the active site, and hence you do not have the regular functioning of the cyclooxygenase. Apart from that, we also know that this chemical is rapidly absorbed in stomach and in the small intestine. We know that its metabolite is salicylic acid. We know that the half life is 13 to 19 minutes. We know that the lethal dose is 1 gram per kg. So we know everything possible about this pure molecule that will be dosed. And based on years and years of experience, now we have evolved clinical trials so that we can catch these chemicals before they are known to, let's say, do harm. At least we know that in the short term. And of course we have lots of post marketing surveillance also to see if there are any long term effects. So there's a lot of thought and effort that goes into these small molecules or larger biologics when FDA approves them and when they go on the market. So, as compared to that, now if we take a look at herbal medicine, I'll take an example of Triphala, which is used, Triphala churna is used. So, it's a combination. So, again, compare this with aspirin, which is one pure molecule. In Triphala, as the name suggests, we have three ingredients. Okay, so now these three ingredients, apart from the fact that there are these three ingredients, they can be given in different combinations for different disease conditions, which makes this again very, very complex. So, if we take just one ingredient of Triphala, which is let's Haritaki, which is Terminalia chabula fruit, we have done some work and we have seen that there are around 120 phytoconstituents just in this one fruit of Haritaki. Okay, so imagine the complexity that we are talking about as compared to this pure one compound of aspirin. So, dealing with this complexity is not easy at all. right? But we need to do something to understand this better. Otherwise, we are never going to be able to get this experience that people have accumulated over years into mainstream acceptability. Okay. So something happened in 2008. A revolutionary paper was published about network pharmacology. So I'll just switch gears a bit and then connect the two. So it was shown that what we think in terms of Western medicine, one drug, one target, one chemical, one target, actually is not true. Okay. So by 2008, a lot of FDA approved drugs were on the market, probably around at least 1,000 or so. And people had information about molecular targets as well. So by 2008, we knew a lot about protein structures. We knew that these structures could be enzymes, receptors, and small molecules, how they bind, inhibit, and carry out their function. So, what was done was it. I mean, this was a really interesting and revolutionary study that this group actually showed that if you make connections between targets, so all the circles that you see on the screen, they are protein targets, okay, which have FDA approved drugs, and the lines that you see are the drugs that connect them. Okay, so a light blue line means that these two proteins share 1 to 10 compounds. Okay, and a black line suggests more than 1000 compounds. So imagine now, of course, these are not just FDA approved drugs but other inhibitors as well. Let's say which didn't make it quite to the market, they were not accepted, but they inhibit. So this is startling, right? So for instance, if you look at the A2A versus A2B, which are GPCRs, okay, adenosine 2A versus 2B, you will see a black line, which means these two receptors share more than 1000 compounds. So the notion that we have that this one pure chemical which is going in our body and hitting just one target may not actually be true, because there is so much interconnection. And that is where this polypharmacology concept came about, that what we are seeing is not probably an effect of hitting one target but multiple targets. And can we do something better? Can we design by principle something that hits multiple? And hence slowly things like polypharmacology concepts started evolving in Western medicine. Okay, so when this network pharmacology and the fact that multiple molecules, multiple nodes, multiple protein targets came about, this was kind of revealing. And you will see, interestingly, if you do a quick survey of PubMed, after this paper was published, if you do a quick search of traditional Chinese or Chinese medicine and network pharmacology, you'll see that boom, publications immediately started rising up by using this concept of network pharmacology, because by this probably you can have a way of saying, hey, this is how our formulations are working, because you are now talking about your approved drugs which are not binding one but maybe multiple things. We already have this. We have in our formulation multiple chemicals, and we are going to use this beautiful network to show how they are hitting multiple targets. Okay. Similarly, Ayurveda also a bit later started catching up, because even Ayurvedic formulations have these multiple polyherbals, right? I mean, they have polyherbals which have again multiple ingredients. So people started using this network pharmacology tool to try and explain how their multiple chemicals might be working. So, in fact, in 2015, one of the first papers of network pharmacology to explain how Trifala might be working came out from the Department of Health Sciences, which is an Ayush Center of Excellence, who are my collaborators in the work that I'm discussing today. So let's spend a bit more time in trying to understand how this network can be used to try and explain how traditional medicine might work. Okay. So let's say I have Trifala, or I have some formulation and I want to understand how does it work better. Okay, so I have ingredients. What I need to do know is what are the phytochemicals in it, right? Which are the chemicals in it? Once I know which are the chemicals in it, I then have to identify, okay, so let's say in my formulation, I have three botanicals or four botanicals, that is ingredient plants. Like in Trifala, I have three, maybe I have five, maybe I have two, or maybe it's pure Haritaki. Okay. So depending on what my formulation or what my medicine is, I need to find out which chemicals are there. Okay. And we'll spend more time trying to understand each step. Once I know the chemicals, let's say I have 20 chemicals or 50 or 100, for each I need to identify, okay, this chemical has the potential to interact with which protein target. Okay. Now that protein target can be associated with which disease. So I have chemicals in my formulation, then I know which proteins they can interact with. Then I need to figure out those proteins are known to be relevant for which disease. And based on all of this, then I can make a network and try and say, hey, this is how my formulation is working. So let's go over this step by step and understand that as of now, the way this is being practiced is that one has to do this manually, right? So each step is going to be done manually, step by step. So the first step, like I mentioned, is that we are going to identify phytochemicals in the formulation. So you have multiple ways of addressing this. So if you have access to experiments, then the best way, of course, is to take your formulation and do an extract and use some kind of experimental techniques like HPLC or some mass spec methods or NMR to try and identify what are the ingredients in the formulation. Okay, so some people like to do experiments themselves to try and figure out what are the chemicals. Some other people say that all these individual constituents have anyway been studied and their metabolites, someone has already tried to extract it. So, can I directly take input from what people have previously published? For instance, I may want to rely on literature, like I'm showing you a screenshot of a paper which talks about assessment of antidiarrheal properties of aqueous extract and soluble fraction of Terminalia chabula fruits. Okay, so in this paper, they have already done an extract and characterized what is there in the fruit, and there are several such papers. So if there is a huge body of literature, then maybe it's not necessary that I reinvent the wheel and do experiments again. But I can depend on what others have done and start by taking chemicals from their published work. Very recently, there is also a database that has been developed by the Mathematical Institute in Chennai, where they have developed this Indian medicinal plants phytochemistry and therapeutics database called IMPAT. So here, what you can do is you can go and browse for the plant of your interest, the plant part. Okay, like I'm showing you here. So, for instance, you can say Terminalia chabula, and what are the ingredients, let's say in the bark or the fruit, and then you can just take this information. Okay, so what you need to do as a first step, like I said, is you need to identify all the phytochemicals in a formulation. Okay, so this requires quite a bit of manual effort where you have to know at the end of this step which all phytochemicals are there in your formulation. Now, the next step would be that once you have phytochemicals or once you have these small molecules identified, okay, and I won't go into too many technical details, but so small molecules can be represented in different file formats. So, at this point, it is helpful if you understand what are these different formats, because you will be dealing with different types of databases later to identify targets and diseases which require small molecules in a particular format. Okay, so one needs a little bit of information here when one compiles these small molecules. So either you can do it in the form of an SDF or SMILES or INCHI format at one place, right? And then you can move on to the next. So let's assume we had the first step where we enlisted all the small molecules and we saved it at our end. Then is the next step, that is, I have chemicals. I want to identify which targets in the human body it may be working with. Okay. Now, this step can be again done in two ways. One is I depend on known association based on experiments. So, several years ago, maybe over a decade ago, there were these large molecular screening initiatives that were started, right? High throughput screen, robotic screens, where then it became possible to screen millions of compounds against different targets. And there are several databases like PubChem, I'm mentioning PubChem here, also Campbell, for instance, which captures all of these bioactivities. So, for instance, somebody has done a large scale screen of a particular GPCR, let's say the beta 2 AR with 1 million molecules, and all the entire assay is available, for instance, on PubChem for anybody to view and download, right? So, there are so many such bioactivity informations across these databases like PubChem and Campbell. Similarly, there are other databases like Binding DB, again, which has a lot of information, experimental information on small molecules as well as their targets, right? So there's PubChem, Binding DB, BioGrid. Of course, there's PDB, which has structural information, co-crystals, that is, you have the target as well as the small molecule, and you have Drug Bank, of course, which is solely for FDA approved experimental, investigational, and all those kinds of drugs. Okay, so if one wants to may build a genuine model based only on what is known experimentally, then in your first step you have compiled your small molecules. So you can go and check if any of such experimental associations are already there in the database, right? Now, quite a lot of these phytochemicals have already been studied in pure form and tested across many targets. So it's quite possible that in your formulation you may capture several of these already in some experimental assay. So then you have a lot of proof, a lot of confidence that this connection that I'm trying to make is valid because it has been shown experimentally. I'll just show a screenshot of one bioassay result, let's say for a small molecule called chebulagic acid, which you can find in many herbal products. So, for instance, this it has many bioassay results, so you can download and you can see immediately that uh, right on the first screen, I see that it is active against this polyprotein for SARS 2, and it has been documented with an IC50 of 9 micromolar, right? So you can kind of do such experimental mining. Now, in the case that you want to go a bit further, because there would be limited information about such associations that have been proven experimentally, then you want to find out predicted targets. There are several methods by which you can do that. So I won't go into the great technicalities, but again, one has to remember that these are predictions. So you can do a similarity search. That is, you have a small molecule and you query your database saying, is there any other similar compound to my query which has a known target? Okay. Or that becomes a basis for similarity search, or you do something called as inverse docking. That is, you have a small molecule and you can use tools like ID target and say, this is my small molecule, dock it against many different proteins and say who is the possible target. Or you can do something like a reverse pharmacophore mapping using a tool like Farm Mapper. But I would say that what you would get from here, one has to really think twice because these would be predictions. So one has to have a very good confidence to include that in the model. Okay, so what we did first is that we compiled small molecules, then we compiled targets of these small molecules, and then we want to make associations with the diseases. So there are several databases that catalog associations between gene products as well as diseases. For instance, DisGeneNet is one of them, which makes associations between genes and diseases based on literature. Okay, so they gave a confidence score. So they've scoured lots of literature, and based on how often these are associated together, they give some confidence that this gene is involved in this particular disease. Then there is a therapeutic target database which enlists approved drug targets. So, whatever drugs have been approved by the FDA, what are the targets, protein targets that are known? Okay, those are kind of enlisted here. And then you have gene cards, which has lots of information about gene, protein, disease, and so on. So you can query such databases then to make a connection between genes, gene products, and diseases. So hopefully now you have understood all the three steps. That is, how do I try to explain how my formulation is working. And I think a lot of people know this already, because network pharmacology now is becoming very common practice and common information to people who are aware. Okay. And then what you do is you make a network. So you can make network in software like Cytoscape, for instance. Now this one is showing a network from Trifala. So it has the three ingredients shown in square. So there is Terminalia cebula, Terminalia bellerica, and Amlaki officinalis. So you can see the chemicals associated with them, phytochemicals, are listed in gray, and the ones that are in diamond are the targets. Okay. So you can see the lines that are there between the squares and the circles. That means those are the phytoconstituents associated with that botanical. And then you see something in the diamonds. So in the diamonds, those are the targets, and there are some letters. Okay, so whether that connection is an inhibition, it's activation, or unknown mechanism. So you can start making such networks, and further, then you can add a layer of disease. So you can say, okay, this is my disease. For instance, one is cancer, one is cardiovascular. So whatever targets we got, we can classify them under heads of different diseases, and say, okay, so this is probably how my network is working. Now, the way this is commonly practiced right now, okay, is that each of these steps that I took time to explain, one has to do this manually. Okay, so it can take anywhere from, if it's just, let's say, one botanical, it can take anywhere from 1 month. And if it's a very complex formulation of 5 to 10 botanicals, so where each will have, let's say, 20-25 chemicals, and it involves a lot of manual effort in trying to create all curate and create lists, and then so chemicals, their targets, what diseases, and then play around with cytoscape to make a network. Lot of manual intervention is required. Okay, so at this point what we thought is that, can we automate this? Okay, so what we have actually done in our work, and we have filed recently a copyright for this. So we are trying to automate this entire process. So what takes, let's say, months of time, I'll show you. We can do this in minutes. Okay. So this is completely Python code, and what we do is, as input data, we need the list of chemicals. So there is some still manual intervention here required, which again we are trying in our second version to automate this as well. So as of now, the user has to give a multi-compound SDF file and a list of disease. So for instance, I want to figure out, okay, how is Haritaki used for rheumatic arthritis or rheumatoid arthritis, right? So to my program, I need to compile in Haritaki fruit what are the different chemicals. I put them together in the form of one SDF file. That's the only manual intervention I require right now. And as input, let's say, as of now, I say only arthritis, but it's possible to give a list of diseases as well. So like we saw, Ayurvedic formulations or any traditional medicine formulation can be given not for one but for multiple diseases, right? So you can give these two as input. So at the back end, what our program does is it will separate each of the molecule in your multi-compound SDF file and converts them into their smiles and inchi counterparts using RDKit. Okay, and why we do that is because right now at the back end we are querying three databases which have experimental information. So the first one is BioGrid, the second one is Binding DB, and third is PubChem. So, and each database has a different requirement. So, for instance, for BioGrid, you need compounds in the form of Inchi. For PubChem and Binding DB, you need Smiles query. Okay, so at our program converts that, queries it, and retrieves again the results will be in different formats. So from BioGrid we get gene name directly, from Binding DB we get protein name, so we convert that into gene name, as well as from PubChem we get a gene ID which is now converted into gene name. So at the back end, our the code that is developed does a lot of search in the experimental database and comes up with a name, a gene name Okay, so you have given a multi-compound SDF file, and internally now our code has created all the genes which are targets for the proteins that are given as input. Right now, all of these genes, they are queried against DisGeneNet database. As of now, we are querying only DisGeneNet, which is downloaded locally. And what we do is that for each gene that we have, we find the disease association. So now we have many diseases. We have, so let's start again. We have many compounds. We found the gene names of all the targets. And for each target, all the diseases they are associated with it. Now we don't need all the diseases. So we filter it by what the user has given as input. Okay, so we then filter it down only to the compounds the user has given. Internally, we have found out which targets are known experimentally and the disease input that the user has given. And then we convert this by code into a cytoscape type format directly in the form of a network, a very basic network, which then the user can play around with and beautify it. Okay. So as of now, we give three different output formats: the CYS cytoscape format, PNG, and as well as an output file. Of course, we'll have more and more revisions as we make this available to our peer community. So our hope is to release it as such. So the user will have an interface, and we are working hard to get this soon to you all, where the user just has to upload the phytoconstituents in the form of an SDF format, enter disease names and click submit. And what, like the I said, the user will directly at the click of a button get a network. And depending on how complex the input is, that is, if you give just one phytoconstituent, one disease, probably you'll get an output in a minute. So we have run different case studies, and I'll just present one. So today's focus is not on discussing the biology, but rather to present this automation that we feel is going to help a lot of people, traditional medicine practices around the world, at the click of a button to get a quick idea of how, molecular mechanism, how the formulation might be working. So we took one botanical, that is Haritaki, and compiled 120 phytochemicals. And we saw that not just impact, but we also scoured literature and different other sources, and we found 120 phytochemicals associated with the fruit. We talked with a lot of different practitioners. We also went through literature and saw that Haritaki is prescribed for so many different diseases. Of course, as of now, we haven't added the layer of the medium. That is, it can be given with ghee, it can be given with milk. So, what role does that have? Let's talk a little about that later. But we found around 37 diseases and broadly under three categories. So we gave all these 37 diseases and 120 phytochemicals as an input. And when we run our code, we see that we get an output network in just 40 minutes. And this is a very simple machine. It's like a laptop which has Windows running. It has 16 GB RAM and let's say 8 i5 cores, right? So as compared to several months of manual effort for curation, we think that, we are going to create significant, we are going to save time of many researchers by putting this out there. So, just to show you that in our case study of Haritaki, we capture around 4,240 unique interactions, and these are all experimentally valid associations between phytochemicals in Haritaki, different target proteins, and these diseases in three different broad categories. Okay, so we are very happy to share that. We are still under the process of copyright. So once that is complete, we'll be able to release this. But of course, I would like to say that this is a very famous statement that all models are wrong. Some are useful. So network pharmacology is not a be all end all, right? Because there are so many things, so many nitty gritties, minute things we do not really understand. So this is really going to be a first step. For instance, like I said, in Ayurveda, we have Haritaki, which is given with ghee for something, with milk for something, or with water for something. We still don't understand that, right? And there are still so many things that we also know are limitations of our model. So, for instance, Right now, what we are saying when we build network pharmacology model is that you go and compile all the chemicals. But like I said, the composition of the botanical might change, so we need a lot of standardization there. Experiments have limitations. So when I say I'm going to compile all phytochemicals, really, have I possibly captured each and everything that is there? Next is that why are we only considering small molecules? What about peptides? What about other things that are there as an ingredient? Might it not have an effect on the body? When we are going to build this network model, we need to go a bit further, a step further, and we also need to ask our question that all these chemicals that we are saying, they are connected to these targets known experimentally, but really, when given or when taken and ingested, are they really absorbed? At what concentrations will they be found in the blood? So this is all, of course, needs to be worked out. And I think we are invested in this, and we want to take this a lot further and work on this so that we really can, understand how these things work. Similarly, when we are making connections, we need to understand which connection is actually going to have a physiological effect. Okay. So there are so many limitations right now, but what we think is we have made a first step towards helping people, at least at the click of a button, they can get a beginning of a network, and it'll save them months and months of manual effort. And of course, what happens is when a person is doing something manually, we can make errors, right? So maybe, oh, I missed this paper, or I missed this assay, I missed compiling this information. But when it is there in the database, a code is not going to make this error. Okay, so we feel that we are going to help traditional medicine community a lot. So for this work, I would really like to thank our collaborators, Ayush Center for Excellence at Savitribai Phule Pune University. They are my neighboring department. So all of my colleagues there, Dr. Swapnil, Dr. Girish, Preeti, and Akash, they have helped us a lot understand a lot more about what is Ayurveda, Ayurvedic principles, network pharmacology, how to build these. At the center, I would like to thank all of the master students who have played a very important role in development of this methodology. So initially, it was started with Sakshi, who tried to first do this in Perl, and we later realized that we need to move to Python. So Isha and Pulkit have been playing a major role along with Aditya to convert this into Python, and the bioinformatics center, of course, for infrastructure support, without whom, of course, this would not be possible. So like I said, we hope to release this in the peer community very shortly, and we hope that this will be very helpful for those who are trying to understand molecular mechanisms of how traditional medicines work. At the end, I would like to quickly say that I come from Bioinformatics Center. We are a center of excellence from DBT 2002 onwards. We run two courses, masters as well as PhD. We are also well known at the national, international, and state level. So, if anybody, any students are interested in joining us, please, you are most welcome. And with that, I would like to stop here and thanks Altem once again for this chance to present our work. Thank you.
Thank you so much, ma'am. It was very informative and knowledgeable session, and many congratulations for this nice work. Actually, ma'am has very well explained the step taken in the network pharmacology, how we can go for the network pharmacological studies, as ma'am has shown that how ma'am has developed the tools to to conclude the conclusion of using the polypharmacological herbs basically. So I'm sure it was very enlightening for my viewers. So let's take some questions. Okay, can we blend traditional with other synthetic molecules to study the pharmacology?
Right. So as of now, in fact, in practice, what people are doing is they take FDA approved drugs as well as they take herbal supplements. So people are already blending, and but like I said at the beginning of my lecture, we need to understand these associations better. Now, even between two FDA approved drugs, there are a lot of studies going on to understand how whether there's any drug-drug interaction. Similarly, when we take a polyherbal, it's very essential to understand whether there can be any deleterious effects between the herbal constituents and the medication that a person is taking. So, yes, in the future, what we want to do is we want to have integrative medicine where we can have the best of both worlds. But I think it needs a lot more scientific study and understanding before we get there. That's
Very true, ma'am. Yes. Thank you. Next. What databases sources provide comprehensive information on traditional medicines, their chemical constituents, and biological activities?
okay. So, if you're talking about Indian traditional medicines, then the Ayush Ministry has a very informative portal called TKDL. So, I would recommend this person can definitely go and browse there. Apart from that, there is a lot of digitalization of Ayush, and again, you will, if that's the type of medicinal system, that traditional system they are interested in, you can look at databases that have been developed by the Ayush Ministry. So that would help them. But as far as chemical constituents and activities go, then you need to do some manual effort still. So there may be a lot of listing, but not necessarily the constituents. So like one says, maybe there are a listing of formulations. Then you need to go and figure out in that formulation what are the botanicals. Then you need to go to databases and figure out what are the chemicals in it and the activities. So then you require network pharmacology, which is a manual effort.
Okay. So, what algorithms and methods are suitable for constructing interaction network between bioactive compounds, targets, and diseases?
So one of the popular software that people are using a lot these days is Cytoscape. So I can recommend that you can look at Explore Cytoscape. So Cytoscape is a software where you can do any type of network, but you can use any other software as well. You can use R or any other different methods as well. So anything that can plot connections, like I said, Cytoscape currently is one of the most popular, but there are several others also. So yeah.
So I think this is the same what ma'am just has
Yeah.
told. How we can generate the network pharmacology.
Yeah, so I think the entire lecture was about that. So, like I said, if you know the formulation, you need to first identify in the formulation which are the botanicals. From the botanicals, you need to identify the small molecules. From small molecules, identify which targets they work on and what diseases they are involved in. So, there are several databases that you have to do cross connections and then finally plot it together. So, that's how you generate network pharmacology.
Okay. So so thank you so much, ma'am, once again for giving your time and for such a great presentation with the novel information what the people are looking into it. It was a pleasure to have you with us today, ma'am. So
Thank
thank
you
you
so
so
much.
much, guys. Stay tuned with us. Please subscribe us, like us, and follow us for upcoming events. Let's connect and grow together. Thank you. Thank you.